Greetings to one and all. I am Bhadragiri Hima Bindu, a PhD research scholar from ICRISAT working on the topic allele mining of novel resistant genes for leaf rust and dead leaf spot in groundnut. Groundnut or peanut is popularly known as king of oilseed crops. It is originated from South America and grown in countries like China, India, USA, Sudan and Nigeria. In India, the low productivity of groundnut is attributed to several biotic and abiotic stresses. Among the biotic stresses, late leaf spot caused by Nothopacillora personata and rust caused by Puccinia arachidis are the most destructive. These two diseases occur together and cause a yield loss of about 70%. They even deteriorate the quality of fodder. Hence, it's very important to develop disease-resistant varieties and identify the genomic regions and genes responsible for disease resistance. Keeping this in mind, a susceptible variety TAG24 and a resistant variety GPBD4 have been crossed to develop a real population. The resistant source of GPBD4 is Arachis cardanaceae. The real population is phenotyped for several seasons and is also genotyped. Utilizing the phenotypic and genotypic data of this real population, a QTL mapping and QTL identification has been taken place. The results reveal that there are some important genomic regions on chromosome 2 and chromosome 3 which are responsible for the disease resistance of both these diseases. So, the QTLs on chromosome 2 and 3 are later sequenced and important genes have been identified. The genes identified are purple acid phosphatase, leucine-rich repeats, pentatrichopeptide repeats, serine threonine protein phosphatase, etc. And the SNPs from these important genic regions have been identified and Cas markers have been developed from these regions. And the developed Cas markers have been validated in several diverse germplasm lines. So these Cas markers can now be utilized for marker-based early generation selections. And these alleles and SNPs from the validated Cas markers have been utilized for allele mining of LLS resistant alleles of Arachis cardanaceae with the other wild sources. And we have observed that the allele of Arachis cardanaceae is different from the allele of the other wild sources. And so we have identified that there are new regions which are responsible for resistance other than the so far identified alleles of Arachis cardanaceae. So in order to identify the new alleles, we have developed the introgression lines and evaluated them for LLS resistance. And we have observed that the introgression lines are performing even better than the GPBD4. So what we have concluded from our study is that these introgression lines can be directly included in the breeding pipeline or can be used as the sources of donors for development of the disease resistance, or they can be utilized for identification of the new candidate genes.